So on behalf of the Scientific Strategy and the Cohort Enhancement Working Group, we are pleased to present on the metabolomic pilot project, which is entitled High Throughput Metabolomic Biomarkers Measures in Diverse Ancestries. And my name is Hákon Hákonarsson, and I'm at Children's Hospital of Philadelphia, and with me today is Dr. Aras Irimadi, and he is a member of the IHCC as myself, and in charge of the Golestan cohort study. Next slide. So I was just going to present a handful of slides that will give us a little refresher on the cohorts and the phenotypes behind this metabolomic projects. And then Aras and I, we're going to lead a discussion session in terms of the application and utility of the data and where this IHCC funding is taking us. Next slide. So to refresh on the program, this started as a pilot program in Q1 of 2021. And there were 4 sites that got funded by the IHCC. And we elected to partner with a company in Finland, which is called Nightingale. And Nightingale has a panel of metabolomic biomarkers. This is highly enriched in lipid and autoimmune and autoinflammatory biomarkers. And the goal was to generate metabolomic data across multiple diversity cohorts, mostly from low middle income countries. And in Q3 of 21, the data from the initial 5,000 subjects that were approved for funding for this program, they got released from Nightingale. And we have been doing analysis, and the pre-set analysis plan was that each cohort would do their own analysis. And then we were going to do a meta-analysis across all of the cohorts, across phenotypes that we could share among the different cohorts. And in Q2 of 22, actually the first metabolomics paper got published from this data. I'll mention that a little later. Actually, June, pre-publication in June of 22. The South Asian cohort led by Dr. Butterworth also has a reach through actually into a huge sample set from the UK Biobank. So this funding enabled that access, both the UK Biobank and the Wellcome Trust. And there are several papers from this pilot project work that are currently under development. Just this past few weeks, we got 2 more cohorts added to those 4, both from low middle income families, Chile actually and Uganda. Next one. And so the initial projects, the cohorts, first project, Adam Butterworth, this is a South Asian cohort, 1,500 samples. Andre Brunori for the ELSA Brazil cohort of 1,000 samples. Aras Irimadi with the Golestan cohort, and myself with a cohort from Children's Hospital of Philadelphia, but this was purely African-American children that we used for this. So total of 5,000 samples. And again, Sandra Cortes for the Chile cohort was just recently approved, as well as Segun Fatumo for the Uganda genome source with 2,000 samples each. So we will have 9,000 samples total with biomarker data coming from these metabolomic studies. And given what we've gotten out of the initial analysis of individual cohorts, I think this could be actually a fascinating project when we finalize the meta-analysis. Next slide. And phenotypes, they were different between different cohorts, but obviously there are also phenotypes that can be shared, as I will mention later. The CHOP cohort was largely focused on autoimmune and autoinflammatory diseases, as well as sickle cell disease and asthma, all in African-American individuals. The South Asian cohort from Adam Butterworth was on diabetes, The Golestan cohorts with primary emphasis of ischemic heart disease with RAS, and the ELSA Brazil cohort was broad-based multiple phenotypes. And the new sites are currently compiling samples, and they were going to select samples that at least overlap with some of the phenotypes we have here, plus potentially some new ones. Next slide. And so just to mention for the South Asian cohort with Dr. Butterworth, the 1,500 samples that he got funded and included sort of gave him access with this pilot data actually to the UK governmental data set. So that was a huge scale up with access to 75,000 samples. And he brings this into the IHCC network. So this is a really powerful resource as well. Next one. And I believe we are going to go to the next one, just touch base on a couple of papers that are sort of one published and one in press currently. This is on the asthma cohort and publication by Dr. Q et al. And this is really showing metabolomic profiling in sort of a sizable cohort of asthma patients, several hundred individuals. And what is well known and characterized is that obesity is a comorbid phenotype that commonly is shared with asthma. And vitamin D deficiency has similarly been shown to be an issue. No one has really published on that in African American individuals, which is sort of the lead patient population in this cohort. But you can see here from the graph that there is a considerable larger proportion of individuals with obesity and vitamin D deficiency, either or both, in the asthma cohort, which is quite interesting. There are also results from the other metabolomic measures and citrate, ketone bodies, including acetoacetate and beta hydroxybutyrate, and the amino acid histidine and glutamine. They were significantly reduced and deficient in the asthma cohort. This is totally new information. It's not been published before. It could be very important for sort of, cell repair, because in asthma, when you get airway inflammation, you get a massive sort of breakdown of the airway epithelium, where these molecules are critically important for the healing process. Next one. And this is similarly a paper which is currently in press, and this is on sickle cell disease, showing sort of, multiple types of biomarkers. You can see that there are 13 groups of biomarkers that show statistical significance. And this is all corrected and controlled for age and sex. Most significant are 41 biomarkers for lipoprotein subclasses that are decreased in patients with sickle cell disease. And you can see the correlation coefficient here is quite substantial and significant. And there are also specific patterns of hypolipoprotein and hypercholesterolemia and lipoprotein subclasses that are vastly different and reduced in the sickle cell patients. This does not include very low density lipoprotein and not triglycerides, but so many other lipoproteins are significantly reduced. Next slide. In this patient population. And you can see here, without sort of going into details, that free fatty acids, total fatty acid, short fatty acid, PUFA, omega linolenic acid, as well as decreased plasma phospholipids are all seen, which are very obviously similarly important in cellular function, cell membranes, development of the brain, and alike. So this is clearly giving us new data that have never been sort of, reported for these populations before. Next slide. So this takes us now sort of to the discussion section, and we were just gonna sort of go through sort of, what are the value of these data, how can we optimize the utility of this, and vice versa. And I was going to ask Aris to sort of tell us a little bit about his experience and where he stands with his analysis on the Golestan cohort. Aris.
Okay, thank you very much, Hakan. It was a very good summary of the work, and I'm very happy to be here talking about one of the projects involved in this effort across the IHCC, and which Hakan initiated and invited us to join, basically. So because I presented before, I don't have slides, but I will just talk about the the basic of what our cohort had and how we participated in this effort. So the Golestan cohort study is a collaboration between the U.S. National Cancer Institute, where I work at NIH, and the Tehran University in Iran, and the International Agency for Research on Cancer in France. It's a WHO-affiliated institute. And this is a cohort of more than 50,000 individuals from northeast of Iran, where they have where the rates of esophageal cancer is high, but we are very also interested in studying cardiovascular diseases, as many other parts of the world, because it's the main killer and the main cause of mortality and morbidity in this region. And when Hakan invited us, actually, we were in the middle of doing case cohort analysis within the cohort, where we had selected about 1,500 cases and controls and reference cohort of people who had died of ischemic heart disease, and a reference subcohort to compare. And we were testing a number of biomarkers, particularly biomarkers of exposure to smoke-related exposures and tobacco biomarkers with CDC, the US CDC. And this might be a good example of how you can really use the opportunities that come along for a cohort from an LMIC, although, well, we have the support of NIH, but basically the field work and everything is done in Iran, which is an LMIC. So we had this infrastructure, we had designed this case cohort study, and as soon as this opportunity came up, we actually added this, the metabolomic analysis to whatever the biomarkers we were already measuring in these individuals. And because the samples were stored in Lyon, it was easy to ship them to Finland, where the lab is. And so we have the results back. It's we haven't been as proactive as Hakan's group, but we have now the data on about 600 cases of death due to ischemic heart disease, mortality in the cohort, both smokers and non-smokers, and about 500 reference sub-cohort, which is a great opportunity to analyze. And also, I'm involved in one of the cross-cohort analysis. We plan to also look at the obesity across all the cohorts in this pilot project. So this has been a really great opportunity for a cohort from an LMIC and low and middle income countries. It was very difficult for us to secure funding for metabolomic study, the omic research in general, and the IHCC platform provided this opportunity to really add something to the data we already have, and to add to the studies that we had designed before. And I really think that this is one of the added values of being in this consortium, and how it can really enrich the data available for many cohorts that may not have the infrastructure or the budget or the basically the infrastructure to do many of these studies on their own. And yeah, we look forward to also welcoming these new cohorts, which really add to the diversity of the project in general.
And thank you, Aras. And we certainly hope that these two cohorts from Chile and Uganda are not going to be the last cohorts coming in. We are hoping that we will be able to continue to get pilot funding and grow that funding into bigger cohort measures, because we can see the added value of having metabolomics, which we didn't have before, most people didn't have before, and it adds substantially sort of to the overall omics data, genomics data, opportunities for phenotype and PFAS type measures to use these metabolomic levels, quantitative traits in various genomic analysis. And I am absolutely convinced there's going to be a really large number of good quality papers that are going to come out of this work. And you know, most importantly, the meta-analysis papers that we sort of, you know, will be doing. I think we are sort of showing that more on the next slide, but I just wanted to stay on this one and sort of, you know, go for a few minutes, sort of, you know, through these points we raised in the discussion session in terms of how can we maximize the utility of IHCC funding. Well, I mean, you know, we sort of, you know, I think have made it in two venues in terms of enabling funding of cohorts that would have had difficulties getting access to these type of data generation. But also this has influenced and affected the price on these measures, and it's coming down significantly. And so we can now, with actually it's very comparable funding from before, we can now offer cohorts many more samples than we could early on. So it's a great sort of, you know, venue to move this forward. And then in terms of sort of major issues to consider regarding data analysis, I mean, obviously this is all data that we have shared. We have shared this with the IHCC. Anyone can have access to these data, but you know, there is obviously sort of similar this to NIH guidelines and rules, sort of a little bit of a protection for us to sort of, you know, do the first pass analysis. But anyone in the consortium can have access to the data, can leverage this for any data they may get funded either through IHCC or through other mechanisms for these type of studies. And so clearly an opportunity to expand the program, both with respect to funding and phenotypes, and sort of, you know, if there are other omics data available on these subjects who sort of, you know, are submitted for these type of studies, that obviously is an added value for any data sharing that can be done across the cohort. So, Aras, I will give the words to you if there's anything you want to add to it, or if not, then we just move to the next slide.
Thanks, Hakan. I can see that there are questions in the platform. Am I right? Are there?
Yeah. Any? Do you want to start? Should we maybe move to the next slide and start on the questions, because some of so I am not seeing that somehow in my chat. If you see questions, yeah.
Okay. Yeah, I saw the questions in the other platform. It says there are two questions. One says, can you comment on sample quality that you experienced in this project for metabolomic analysis? Any lessons learned? And the other question is also related to samples, which says, which samples and what volume is used for metabolomics assay? I'll try to start answering, and maybe Hakan would like also to comment on that. So the samples used were plasma. So they can work with serum as well, but they prefer plasma samples for these assays. And the volume is really minute. The Nightingale platform uses about 100 microliters. So what happened in the case of Golistan, we were actually keeping these samples in IARC in straws. And those of you who are familiar with the straws, these are very tiny tubes. Each of them had about 200 microliters in them. So we shipped them there, and they were very conforming in working with the straws, and they returned the half of the sample that was not used. So very little sample is required. They are very expert. The lab is very expert in working with different types of the sample platform, sample storage. They have a lot of experience working with UK Biobank, etc., with handling these samples, etc. And we didn't really face a lot of challenges, and there was good quality control on the data, and I think we didn't face the specific problems, but Hakan may also would like to comment on that.
Yeah, no, I agree 100%, Aras, and similarly, I mean, we had samples that we collected back in 2006 when we started our center. There were multiple samples from the early days. They were all of great quality, gave great data. And, Nightingale actually gives you the offer to submit a little bit more sample and pay less, or you can submit as little as 100 microliters. And it's just a couple of more dollars per sample. And most people prefer, and that's what we all did for this first sort of, data set. But the data is of really good quality, excellent distribution. So no matter what measures we've used on sort of, looking at the quality of the data, it's really excellent. So are there more questions, Aras, that you?
There is one question. There is another question also in the Q&A here. Yeah, how much additional data, non-metabolomic data, is shared? How do you manage data access agreements? Some countries have restrictions on sharing personal data. This is something that we have discussed a lot because of the mandates for sharing of omic data, and we're, as Hakan mentioned, we share the basic data on individuals. For example, for our cohort, unfortunately, the consent that we obtained does not allow us to go beyond the basic like things like age, sex, posting on public website. But we definitely, the group as a whole, and I'm please Hakan correct me if I'm mistaken. The group is open to people proposing to work with the data and suggest proposals to use the data. I mean, other than the public platforms that we are obligated to post on, obviously.
Yeah, no, I totally agree, and we follow the IHCC data sharing policy by every measure for this data set, and it's voluntary in terms of I mean, we have GBAS data on these patients. We are happy to share those. We have permission to do it, and so we are going to do that as well. But there are different sort of restrictions by different cohorts. So those who can, we hope they do, and where there are regulatory restrictions, I mean, we all respect that. Yes, I know there are a couple of more questions coming in, but just so we can go over the last slide very quickly. And then Aras, maybe you take the questions. The data sort of dictionary for sharing, we want to share demographic data, phenotypic data, and the metabolomic data, and any available other omics data that is allowable, permissible for sharing. And we are working hard to get additional support for other sites to expand and grow the program, because we think this is going to be very valuable. And we are working towards the common papers, should be plural, across all cohorts on sort of phenotypic traits and diseases that we can share. And obviously BMI, everybody has a sort of height and weight on individual blood pressures measured almost every clinic visit, and most cohorts have some laboratory measures, hemoglobin, platelet counts, CBC on or chem panel or something, or other types of tests. This could be radiology, imaging, pulmonary function testing, or sleep studies, or echoes, or EKGs, etc., etc. So there's substantive opportunities to sort of do this. I know we are getting close to time, but maybe Aras, you can take a couple of more questions. I think there are a couple of more questions in the.
Yeah, there is one additional question, which was actually earlier than the last one I mentioned, but I was waiting for you to show this slide. So the question is, how did you observe any geographical differences in profiles that might be attributed to diet and other cultural differences among participants? that may confound the analyses. As Håkan is showing in this slide, we are now working on those common papers which combine the data across these different cohorts in this pilot project, and that's the platform where we can also explore differences across these population, geographic, and as you mentioned, diet wherever available. In Golistan, we have diet, probably on some of those cohorts also we have diet. So that's a very good question, and this is something that we will address in these common papers, common analysis that we will planning to do.
Great. And I think last slide is just a thank you. I believe we are up for time, and unless we are notified that we have more time, we'll have to continue. But this was I think a great program, sort of representative of what IHCC can do. That is of major added value to so many cohorts that would otherwise not have the opportunity to have access to these type of data.
Exactly, I second that, and this has been a great experience working with this cohorts and this IHCC platform. Thank you, everybody, and thanks, Håkan.
Thank you, and have a great rest of the session. Thank you.